welcome everybody, and welcome Niels to our webinar this evening. Just to give you a little bit of background, as a project which supports commercial horticultural growers in Wales, we've set up various grower networks, and one of these being the Tyfu Cymru Vegetable Network, which is managed for us by Debbie Hanley. Debbie's joined us this evening, and prior to COVID, she went round and spoke to growers extensively. With the information that she gathered, she put together a full program of events and CPD training. One of these events was going to be a face-to-face with Niels Corfield. But in this new way of working, we've managed to convert this training delivery to an online webinar. So we're delighted to welcome Niels, and we're very interested to hear what he's got to say to us tonight on soil health. But just before I hand over, just to make sure everybody's happy with the technology and the procedure tonight, we've put our housekeeping rules in the chat. If anybody's new to Zoom, if you move your cursor down the bottom, you should be able to see the chat icon. If you click on that, you'll see the housekeeping rules. And to the left of that, there's a Q&A box. So we'd encourage everybody to, if you want to ask a question, do it through the Q&A box. Write the questions as Niels is going through the webinar and the information, but the questions he will answer at the end. So we're expecting to run for about 45 minutes and then the Q&A session at the very end. So I will now hand over, and thank you once again for delivering this evening.
You're very welcome. So yeah, hopefully you can all see that. So yeah, like was said earlier, I'm delivering this for Tyfu Cymru, and the session's called Soil Health for Commercial Growers, and here's the various kind of links that you can follow up if you need. Otherwise, just a little bit about myself. Basically, I offer advice and education for growers and farmers, helping them to apply soil health practices in the field, setting up small-scale on-farm trials, and carrying out monitoring and training, as well as training you guys to carry out your own monitoring. So what I would like to do sort of initially, just get the chat window open, there we go, yeah, so kind of talk about this idea that, you know, if there is one thing that we could get growers and producers to agree on, I know this might be a controversial statement, but if there is one thing that we could all agree on, I think it's this, and that your land is in best heart the first year out of grass, basically. You know, obviously, sort of wireworm and other kind of like considerations aside, but basically, it's this kind of like idea that sort of underpins what we're going to look at today, basically, and the idea of basically bringing the heart of grass, if you like, to your cropland, but without having to go to grass, basically. Okay, so it all starts with observation, and what I would encourage all of you to do is to get your spade out and take some spade samples, and they're very simple to take. Basically, all we need is to cut like a spit of soil, like a kind of regular square of soil, basically, lift that out, and then kind of break it in half to kind of reveal the interior structure of the soil, basically. We don't want to see any spade marks or any kind of anomalies. We just want to see what the sort of internal structure of the soil looks like, basically. So we're looking at two soils here, side by side, two spade samples. If we zoom into soil 2, the one on the right, you can see it has a sort of blocky, quite a sort of consolidated structure to it, basically, quite sort of solid, if you like. as opposed to soil 1, which has much more kind of open and loose kind of structure to it. And if we zoom in, we can kind of see along these roots a little bit of kind of, you know, sort of maybe rounded or bobbly shapes that look a little bit like mini earthworm casts, you might say, like along these roots here, for example. And otherwise it has quite a sort of open and loose structure. You can kind of see into the soil there, basically. So yeah, if we then look at these soils again side by side, again, the one on the right, quite blocky, quite dense, quite solid, as opposed to the one on the left, it's quite open, loose, and sort of crumb-like, you might say. Yeah, so we probably ask the question at this point, are they the same soil type? So based on just what you're looking at, are the same soil type or different? Well, what we could also say is, you know, are they quite similar or quite different in their appearance? Well, I think that we could probably say that they are quite dissimilar, except that the color is quite similar. So, you know, from that we can probably infer that they are the same soil type. But then we might ask the same that question: are they the same land usage type, basically? And we'll come back to that answer in a bit. But basically, let's look at another couple of soil samples here. Again, sort of on the left-hand side, we're seeing kind of quite blocky, in this case, also quite cracked soil structures, if you like, and also quite pale relative to the example on the right. So if we look at sort of into the sort of interior structure of this soil, we can see a lot of sort of cracked, maybe angular features, sort of seeing jagged edges, some kind of surfaces, for example. And otherwise, these two soil samples are quite different in their color. So the one on the right is quite sort of dark brown, basically. And in this case, we'd ask that same question: are they quite similar or dissimilar in their appearance? And again, probably the structure looks sort of similar, but the color is quite radically different, isn't it, basically, in this case? So again, we'd ask that same question: same soil type. And in this case, as with the one before, they are both the same soil type, but different management. So the one on the left-hand side is conventional arable, and the one on the right-hand side is grass margin. So the final example we'll look at, again, to ask this same question, you know, would we, you know, based on the appearance of these soils, would we say they're the same soil type or different? Again, you know, quite different colors, for example, and then also quite different structure. And again, if we zoom in to the image on the left, we can see some of those kind of jagged edges, kind of surfaces, particularly along this sort of edge here, for example. There's definitely kind of some sort of jagged edges, a lot of surfaces, faces, quite, again, quite solid, quite dense, quite consolidated, if you like. And this is another opportunity for you to get feedback about your soil, basically. So in this case, kind of, I took these two samples with the spade, obviously, and in this particular case, we're looking at lawn on the right hand side and arable field on the left. And again, same soil type, but you can, you know, quite radically different shapes and forms, basically. And in this case, basically, these soil samples were taken from the garden on one hand, kind of a client's garden. And in this case, his garden just ended sort of directly into the land that they owned, one of their arable fields. No hedge, no fence, nothing, just edged lawn, and then arable field, basically. So we can kind of get some feedback when we're just putting the spade in the ground as to the structure that we're sort of seeing or feeling, basically. So in the case of the lawn, that's spaded pretty easily, or at least, you know, nothing to report, and, you know, went in fine, came out fine. However, when I took a couple of paces into the arable field, couldn't get the spade in, literally. Was that different? At which point the client was like, oh, I'm convinced. I wasn't sure exactly what I was supposed to be convincing him of, but anyway, he seemed pretty kind of shocked by that, and as was I. Obviously, then I took a few more paces into the field and was able to take this spade sample, but you can see they are kind of radically different. So in all of these situations, we've looked at the same soil type, but with different management. Yeah. So we can basically say that management is one of the key determining factors of soil health, how you treat the soil, basically. So the next thing we're looking at basically of those kind of soil structures then basically is kind of like zoom in a little bit and ask this question, well, you know, if your soil has sort of angular features, kind of jagged edges, you know, we're seeing fissures, cracks, if it cleaves along fault lines, for example, you know, then basically, you know, it's behaving like kind of rocks basically, or it's looking and behaving like rocks or otherwise like geology. Yeah. So, you know, we're looking at these two examples, you know, we're seeing quite similar shapes here, kind of jagged edges, shear faces, for example. Yeah. And on this one, we're seeing, you know, those kind of fault lines, we're seeing those cracks, those fissures kind of, you know, where the soil basically has kind of cleaved. Yeah. So like slate or, what's it called? Gritstone. So basically, you know, we can say that like if your soil looks and behaves like geology, then basically it is just geology, which kind of makes sense because ultimately soils are just made up of fundamental, or the large majority, 90% of the soil are just made up of ground up rock. Yeah. However, we would want to sort of like differentiate that from topsoil, basically. So subsoil is where it's basically made up almost exclusively of the mineral base material, the parent material, for example, whereas topsoil is that layer of soil that has been acted upon by biology. Yeah. And that's what gives it its dark color, and that's what gives it that different structure, basically. So we can basically say that or if there is one master indicator of soil health, yeah, the one thing that you can look at and look for, it is aggregation or crumb structure. So those rounded bobbly shapes that we looked at earlier, for example, or that might have the appearance of earthworm casts, for example, mini earthworm casts. So what are aggregates? You know, what are crumbs? What is crumb structure? Well, just going to use a baking analogy now to kind of illustrate this point, and basically sort of ask this question, you know, who here makes biscuits, was ever made biscuits or crumble topping? Well, we all know that the base ingredients are sugar and flour. Yeah. And, you know, the sugar particles have a kind of granular consistency to them, quite sort of square or sort of rounded, if you like. And, you know, in terms of how similar each sugar grain is to each other or to one another, for example, you know, how similar is each sugar grain compared to each other sugar grain? Well, in this case, they are very similar. In fact, almost completely sort of uniform, if you like. And the same would be true of the flour as well. Yeah, so you can pass sort of like 99.9% of the flour through a sieve, for example, the odd little bit of sort of husk that made its way through the mill, maybe getting sieved out, but basically, so they're all the same shape and size. So in this case, basically, we would refer to these materials as homogeneous. So each flour particle, for example, is the same shape and size as all of the others. Yeah. So what's the kind of third ingredient that turns it actually into biscuits or crumble topping? Well, that's the fat, isn't it, basically? So you put the butter and you knead it in, and what do you get at the end? You get crumbs or crumble, basically. And in this situation, you know, how has the size and shape of those materials changed? In this case, you know, we've gone from, you know, very regular size and shape to quite dissimilar size and shapes. You know, there's some, you know, big lumps in here, for example, there might be about 12 mil across, for example, half an inch. And there's some in here that might be just like a 1 or 2 mil across. So, you know, basically gone from everything being exactly the same shape and size to stuff being potentially about an order of magnitude difference in size, for example. You know, something like you know, sort of a bowling ball next to a tennis ball, you know, really different in size, and also again sort of very rounded in that structure, if you like. So the same process is happening in the soil, yeah? And in this case, our kind of base ingredients would be sand. So the granular material that's equivalent to sugar is sand, yeah? And clay, if you get dried clay in a bag, how does it come? It's a powder, isn't it, basically? If it wasn't, you send it back basically because it's got wet, you know, and you've been sold a dud. So, you know, in this case, you know, clay is a powder and the sand is a granular material. But the binding agent in this particular situation are things like polysaccharides. And again, just to use a real world analogy to illustrate what this material or what these materials are like, has anyone ever got a slug onto their clothing and potentially not got round to sort of washing off what it leaves behind? Well, what does it leave behind? How does it go on? Is it easy to wash off? And what does it like after a few days or longer? Well, it goes on sort of wet or sort of slimy, sticky, sets hard, and is hard to wash off, really quite difficult to wash off. So what kind of things are sort of similar to that we might know of in the workshop, for example? Well, glue. Yeah, so these kind of compounds are glue-like in their nature, basically, kind of very sort of recalcitrant, go on sticky, and like kind of bind together these soil particles into aggregates, yeah, or crumbs. So it goes from the homogeneous state to the heterogeneous state. So you have like a whole variety of different sizes, basically. And it's these gels and slimes that give the structure to the soil that we all crave, if you like. And I think the most important thing I want you to remember at this point is the word saccharide. Yeah, what is the word saccharide derived from? What's its sort of the root of that word? What it means sugar-like, sugar-derived. Yeah, and what's in sugar or sugars? You know, what are the key sort of elements that make up sugars? Well, it's carbon, hydrogen, and oxygen. So these are carbon-based compounds, long chain carbon-based polymers that are gluing together these soil particles. So in this case, basically, we're starting to kind of bring this together. And our first kind of like sort of key insight, if you like, is that microbes make aggregates. So these are the ones that are producing the gels, the slimes, the biotic glues, the polysaccharides that are binding together the soil particles into those crumbs, into those aggregates. Or actually, in the case of fungi, also wrapping them up with their hyphae, yeah, sort of binding them together with sort of fibers, basically. So microbes are the ones that are making the aggregates. They make the binding agents. Yep. So what's the big deal with aggregates and aggregation? Why do we like that? Well, if we come back to our baking analogy just briefly, you know, when you sort of add the fat to your material, or rather when you put the material into the bowl and pour it in, for example, maybe sift it, how does it occupy that volume? Well, it fills it, doesn't it? It basically completely fills that volume to a level, if you like. When you add the binding agent, when you add the fat, basically, how does it change? It sort of fluffs up, doesn't it? Basically, sort of kind of lifts up. So what, you know, we haven't had much material basically, but actually we've increased its volume. So what have we done there? Basically, we've exchanged matter for air. So we've basically created internal space or pore spaces within the material, and the same thing has happening in the soil. Basically, we've created internal space or pore spaces. Yeah, we also have a nice sort of crumbly texture basically, and with it sort of tilth that again we can all utilize. So if we're looking at aggregation or aggregates in more detail, then here's an example of a diagram on the left where sort of we have microaggregates, which are basically bits of particulate organic matter, POM, bound together with sort of gels and slimes, polysaccharides, for example, and a whole bunch of organisms living in there. And they themselves, in turn, are bound together in larger aggregates that you can actually see, basically. So macroaggregates are made up of microaggregates, but it's all basically the same material, basically. So, you know, what's the big deal with pore space? Why do we like pore space? Well, one thing we gain is we gain improved infiltration. Yeah, so that means that water doesn't puddle on the surface, for example. It's much less likely to saturate these soils. Will move through soils that are heavier, for example. Yeah, so we improve the infiltration rate on heavy soils, for example. We also gain more aeration. You know, that pore spaces are spaces that air can exchange into the soil. Yeah, and do we want air to exchange into the soil? Well, yes, we do, basically. What's in air that we want to get into the soil? Well, things like nitrogen, for example. Yeah, if we want our nitrogen fixing bacteria, our legumes, to be able to do that sort of job of nitrogen fixation, they need to be able to access some blooming nitrogen, you know, so we need good gaseous exchange into the air, into the soil, and that aggregate structure brings that, basically. The other thing that we gain is we gain additional surface area, basically. All of these aggregates, the microaggregates and macroaggregates, basically, they have like a surface area, basically. And this is areas where water films can collect, basically. So we can think of the soil as a kind of subaquatic environment, yeah, and where you have good aggregation, basically, you can improve the water holding capacity, particularly for lighter soils. Yeah, so that that increased surface area also allows soil to hold on to moisture, but rather than it basically just filling up and becoming saturated, you have all these kind of isolated water films that coat all of the internal surfaces, basically, which provide a reserve or reservoir that can then be drawn upon by plants in the season, basically. So if we look at this kind of structure and how that changes the structure, if we're looking on the left-hand side, for example, you know, if we're working with sort of heavy soils, you know, ones that are quite blocky and consolidated, these tend to waterlog and be quite claggy, for example, in the winter. When we increase the aggregation in those, then we make them more free draining and they become lighter. Yeah. And on the other hand, you know, if you're working with very light soils, like very sandy soils, for example, they tend to be quite kind of structureless, tend to be quite droughty and prone to blowing, for example. When we make these more aggregated, or when we increase the aggregate structure in these soils, they become more loamy, yeah, and more moisture retentive, basically, as we saw before. And basically what we gain is a natural fine tilth, the kind of tilth that persists, as opposed to those that are created with this is kind of a biological tilth as opposed to a mechanical tilth, and that lasts, basically. Okay, so we might ask this question now to kind of sort of move on a little bit. Do we like legumes? Do we like the peas and beans? You know, are they good? Do we like them in our rotations? Well, what's good about them? Why do we like them? Well, again, you know, just to sort of like be really explicit, they associate with nitrogen fixing bacteria that bring basically free nitrogen into the system. Yeah. So, you know, we might ask this question, sort of, where does that nitrogen fixation take place? Well, it takes place inside these kind of swollen cysts or galls that grow on the side of the roots, which we call nodules. Yeah, but we might also ask this question then, you know, sort of why? Why are the nodules produced? What's special about nodules? Why does the plant go to all that effort? Well, yes, it's a home for those organisms to live, but the most important thing basically is they create a sort of climate controlled environment or a low oxygen environment. Yeah, so you could ideally, again, we want good gaseous exchange to the roots, yeah, to the nodules, yeah, but then the nodules basically restrict the amount of oxygen that can actually enter the internal space, if you like. So you have an outside and an inside. Yeah, so the outside is bathed in air, it's bathed in oxygen and nitrogen, for example, because there's loads of nitrogen in the air. But there's a restriction on the amount of oxygen that can get in, basically. And that's basically because this compound, or this enzyme, nitrogenase, only works in the absence of oxygen. Yeah, it only works in anoxic environments. So you can produce as much nitrogenase as you like, basically, but it ain't going to produce do any nitrogen fixation unless you exclude the oxygen. So that's what the nodules are there for, basically. And an interesting point to note, and another opportunity for you to sort of do some observation sort of your soils basically, is that you can of your plants and your legumes, dig up some of your peas and beans, for example, and examine the roots, and you can have a look at how much nodulation there is and how big they are, for example. And that's a good sign, basically, if there's a lot of nodules or large nodules. And then you can actually cut them open, for example. And if you're seeing sort of pinky or kind of maroon colors inside there, basically, that means that nitrogen fixing is taking place. And that color, that sort of pinky maroon color, is the color of the nitrogenase. And again, might say, is there anything similar to those kind of colors we might find in the body? Well, it's quite a similar color to blood, isn't it, basically? And that's basically because nitrogenase as a compound is a very similar structure to hemoglobin, except that rather than having an iron at the center, it has magnesium. So yeah, these are more sort of opportunities for you to sort of glean the health of your plants and whether your legumes are actually fixing nitrogen as opposed to whether they should be or could be. Yeah. So then we might sort of like draw a comparison now between these shapes and these structures we've just looked at with the nodules to what we just looked at before. So what have we just looked at before that has a kind of rounded structures, sort of bobbly structures, an outside and inside? Well, basically, soil aggregates or crumbs are directly analogous to nodules. Yeah, they have an outside and an inside. They have a rounded shape, basically, and these then become homes for free-living nitrogen-fixing bacteria. Yeah, and free-living nitrogen-fixing bacteria are some of the most abundant bacteria in the soil, organisms in the soil, but they themselves need a low oxygen environment to produce functional nitrogenase. Yeah, so they do it in the same manner, basically. So we can basically say that our aggregates are like a whole soil full of nodules. Yeah, so all of those kind of non-leguminous green growing plants that are out there in the nature are almost certainly going to be associating with these free-living nitrogen-fixing bacteria. So nitrogen fixation is certainly by no means limited to legumes, but we want to partner with these organisms. We want to give them homes, and we want to give them that soil structure that is a home for them, and we want to make sure they're fed. Okay, so if we want to put some metrics or measures on the degree of aggregation that we've got present, we can do things like the VEST test, visual evaluation of soil structure, bulk density, so how much volume of soil occupies, or how much mass of soil occupies what weight. So lower densities means more aggregation, more air spaces, more pore spaces. We can use the Slake Test, and we'll look at that in a moment. But we can also look at infiltration rates. So infiltration rate is basically just where you take a ring or a tube, basically insert it into the soil, pour a known volume of water, in this case 1 inch, yeah, and then time how long it takes for that water to percolate through the ring and into the soil. So we've got, again, those same soils that we looked at earlier. And on the right-hand side, that soil could infiltrate 1 inch of water in about 220 seconds, which is not bad. It's a pretty good result. So we'd say it's generally quite a free-draining soil. On the left-hand side, though, that soil could infiltrate that same inch of water in either 6 seconds or 25 seconds. So an order of magnitude more rapidly, basically. Yeah. So again, same soil type, different management. And we are seeing a different, we can see differences in the structure. It's more open and loose. And that's also borne out in the way that water infiltrates, basically, because the more pore space, the better the aggregation, the faster the water can move through that soil, basically. Okay. So we can basically say that through all this, there's sort of one key insight that underpins all of this thinking. And that is basically that microbes matter. Yeah. And by microbes, we just basically mean sort of microorganisms. Yeah. So why do microbes matter? What's kind of, what do they do for us? Well, as we've seen already, they structure soil. Yeah. But as well as that, they also feed plants and protect plants. So how do they feed plants? Well, quite simply, microbes eat rock. Yeah. And organic matter to an extent. Yeah. But we might ask this question, what is the soil made up of? What's 90-odd%, it's basically ground up rock and some organic matter, yeah. But basically, what are these rocks made up of? Well, they're made up of minerals, aren't they? Elements like calcium, magnesium, phosphorus, potassium, etc., etc. Yeah, and collectively, what would these sort of elements be known as, like in the garden or on the farm or on the plot? Well, they would be known as nutrients, wouldn't they, basically? So the term nutrient basically is just a fancy word for some element or any element that is required for life, basically. Yeah. So we might ask this question then, are those mineral nutrients that are making up that soil, are they available? Are they plant available, for example? So if I took a cup of water, put some silt in there and drank it down, would it satisfy my appetite? Would I be able to derive much nutrition from that? Well, not much really, negligible, yeah? However, the microbes are able to do that basically, but rather than them having like an internal digestive tract like we do, you know, they can't swallow their food basically. They're basically just lying on their food, lying on their food, kind of like gradually just digesting out minerals from the soil mineral matrix basically, and absorbing them into their bodies. Yeah. So we can basically say, you know, when we say microbes eat rock, they are basically producing enzymes, yeah, that are specially adapted to digest or mine or mineralize, or a better word is solubilize those mineral nutrients basically, at which point they assimilate them into their bodies, and thenceforth they become bioavailable. Yeah. So we might look at mycorrhizal fungi to kind of sort of make this same sort of point, if you like. So in the same way that bacteria and other like organisms, they are also producing enzymes and strong acids that are able to strip out what's it called, mineral nutrients basically, and make them plant available, pass them back up to the plant. But in the same way that the lungs are super efficient at accessing a gaseous nutrient, in this case oxygen, yeah, every time you take a breath, you get the most amount of oxygen out of the air. Yeah. In this case, the plant that associates with that mycorrhizal fungi gains a huge amount of surface area, to volume ratio. So that's what the lungs are all about. Super high surface area, basically, compared to that volume. So each lung basically laid out is the size of a tennis court. So in this case, the mycorrhizal fungi, you know, has that huge distances, like loads of miles of length, for example, of the actual strands, and the surface area, again, you know, when you add it up, is much larger than just the root area on its own. So by partnering the plant with the plants, basically, or rather, with the plant partnering with the fungi, it gets access to low concentrations of phosphorus and water at great distances that it then can utilize for itself. So we can basically say that microbes are providing a bit of a bridge between the mineral nutrients and available nutrients, yeah, through that process of digestion rather than really than decomposition. So the next thing that microbes do is they protect plants. They provide pest and disease suppression, and that's done by being recruited by plants. So the plants actually have an active role where they're able to recruit organisms to carry out tasks for them, basically. So in this case, the different ways that microbes protect plants is they can mask the roots, yeah, so they can basically produce sort of aromas and other sort of compounds that camouflage the roots, and that means that like pest organisms will just sort of motor on by because they can't see those organisms, can't see those kind of target plants, if you like. They also repel organisms, particularly pathogens, by producing antimicrobials. They can exclude them by occupying the surfaces around roots that kind of no room at the end, and they can also predate kind of pest organisms. So here's an example of a root-feeding nematode. Why are they? Well, they're bad. Basically, they're bad because they burrow through the root walls and create entry points for diseases. And in this case, this nematode has been snared by this mycorrhizal fungi, at which point then it will digest the nematode and pass the nutrients up to the plant. So the plant's getting a pretty good deal here, isn't it? It's got its own ground crew, basically preparing soil, loosening it up for nice, easy root growth. It's got its own live-in caterers, its own room service, and its own private doctor. Yeah. Is it getting all that for free? Well, as we all know, there's no such thing as a free lunch. Yeah. So in this case, you know, what's sort of driving that, or what's the currency that's driving that? And basically, it all comes from photosynthesis. So this is one of the kind of the magic adaptations that plants have achieved. So they're able to convert sunlight energy into sugars. So they're able to fix carbon through photosynthesis, at which point they then translocate those sugars down and out through the roots as root exudates. Yeah, at which point they basically become a food source for other organisms. So this is when we come on to the soil health principles to kind of like bring this all together, basically. So we've looked at microbes matter, for example, and now we're going to look at these next few examples. And the first one of the soil health principles is to maintain the living root for as long as possible and as often as possible. And that's basically because we're in a cropping situation where we can't ensure that 365. Why is the living root so significant? Well, basically, as we've seen, roots feed organisms and organisms nourish plants. Yeah. So, you know, this is what those sugars look like. This is what those kind of exudates look like. And what they do is they feed like a kind of a mass of beneficial organisms that live around the roots. In this case, the majority of them will be bacteria, basically, feeding on those sugars. Yeah. And, you know, what happens when you have a whole bunch of sort of like small organisms kind of, you know, feeding on something, you know, their population increases. And then what gets interested in them? Well, predators. Yeah. So, you know, when these organisms like bacteria are predated, when they're eaten by things like protozoa, for example, or nematodes, yeah, they will be kind of an excretion process. Yeah. Because, you know, the byproduct of ingestion is excretion. What do we call excretia on the plot? Well, we call it manure, typically, don't we? And we might ask this question: do we like manure on the plot? Well, basically, yes, we do. But what or why do we like manure? Well, basically, it's because it contains nutrients. Yeah, and what form are those nutrients in? They are in a plant available form. Yeah, that's why we like muck. Basically, that's why there's muck. Where there's muck, there's brass, because basically it contains an abundance of plant available nutrients. Yeah, so the same process is happening in the soil, right in the root zone. They're basically, in this case, you know, protozoa. They're motoring along and hoovering up, you know, tens of thousands of bacteria every day. At which point they basically are depositing or excreting small amounts of plant available nitrogen right in the root zone. Yeah, so you could say that through like a number of intermediate steps, the plant is basically paying for available nitrogen in sugars. It's like, here you go, guys, here's some sugars. I want to get my nitrogen back. Yeah. So if we look at the legume example, that's an example of a kind of closed loop system, basically. So the plant is passing down sugars, photosynthetic products, down to the nodules. Yeah, that's feeding the bacteria that are living in the nodules, and in return, the nodules hand back nitrogen, basically. Yeah, so it's a kind of a closed loop system as opposed to the root zone or the rhizosphere, where it's a more kind of open system and open loop, if you like, or cycle, and that may have a number of intermediate steps. Yeah, but basically we can kind of summarize this whole point by saying that, well, energy in, nutrients out. Yeah, so energy in, or in the form of sugars, because again, sugars contain calories. Yeah, they contain carbon. Yeah, so it's an energy source. Yeah, that is basically driving the nutrient cycle. Yeah, it's the fuel source that goes into the tank that drives the nutrient cycle that then comes back to the plant. Yeah, so energy in, nutrients out. Yeah. So that's when we come on to this idea of finding the best roots, best soil around roots. Yep. So this is an opportunity for you to sort of go out and examine the soil and see what it would look like on your site, basically. But generally speaking, we're liable to find the best soil around roots because that's where the most microbial activity is. Yeah, so fine structures and rounded shapes. So it might be something like this around some kind of cover crop, for example. Again, that sort of fine structure. Or it might be around like a brassica root here, for example. Again, we've got a nice fine structure, nice sort of crumb structure, just sort of natively there. And this is right at the end of the season, you know, sort of April, so it might be in the ground for 9 months. You know, so any kind of structure that might have been gained, you know, during the kind of tillage process, you know, it's sort of long gone, basically. So these are kind of sort of natural tilths that we're looking at here. And again, it's nice, you know, crumb structure. So yeah, it's all good, basically. What we also might be seeing is nice matted roots, for example. Yeah, so where we see these dense matted roots, particularly around young seedlings after transplant, for example, that is indicative of rapid, aggressive root growth, and that's a good thing, basically. So the next thing we might ask the question is, well, we can make a sort of inference that if the healthiest soil is to be found around roots, then the healthiest soil of all will be found around the healthiest plants, yeah? And generally speaking, you know, what are likely to be the healthiest plants on your plot? Well, unfortunately, it's most likely to be weeds, yeah? Or at least currently. So another opportunity for you to gain some insight as to how that soil can look when it's being structured well, basically, is you can lift some of your weeds, small number, just carefully, and examine the roots. Maybe place them down on a tray, take a photograph. And again, we're looking for those sort of fine structures, for example, that small particle size, you know, that kind of fine structure. And then also we might see soil stuck to the root. You see there's some soil that's stuck to this root here, basically. And what's holding that soil on? Well, basically it's going to be those gels and slimes and sticky substances that are binding together that soil, as we saw before, basically. So, you know, we can infer from that that we've got exudates being produced, and that's feeding the organisms, and the organisms are producing the gels and slimes and sticky substances. And when they're a bit further down the line, you might actually see kind of a whole root kind of coated in what we call rhizosheaths. Yeah, and in this case, you know, we've got a whole kind of linear aggregate full of free-living nitrogen-fixing bacteria. And this is basically where those aggregates are born. So you can kind of imagine aggregates as being like carved off the face of a glacier. But in this case, the face is the root surface, basically. So aggregates are born, microaggregates born right next to this, to the roots. And as they get older and mature, basically, they bind on to other ones and then gradually move out into the open soil, basically. So what do we want to then, well, again, so the next thing we'd ask this question, you know, is the living root present 365 days on our plot? Generally speaking, if we're in a cropping system, no. Yeah. So then I also ask this question, does the effect of the living root kick in on day one after sowing or transplanting? No. There's a kind of lag time, isn't there? So what is the kind of key sort of determining factor that dictates how long it takes for that, the effect of the living root to start kicking in, basically? Well, let's look at a couple of soil samples here. So again, same day, same soil, you know, taken from within a few meters of each other. Yeah. On the right-hand side, we've got a herbal ley. Left-hand side, we've got a game cover. Yeah. And, you know, what's the sort of glaringly obvious difference between these two? But on the left-hand side, we've got a lot more soil there, much bigger sort of root mass and soil structure, basically, whereas the one on the right is just pretty shallow, basically. And that's all the soil I could get out. Yeah. And what's the key difference between these two? Well, on the herbal ley on the right, for example, that's all perennials. Yeah. On the left-hand side, that's all annuals. Yeah. So again, sowed the same day, using the same method, basically. So all other kind of conditions being equal, and in that same period of time, those few months, basically between germination and sampling, you know, the one on the left, the annual mixture has massively increased or improved that soil structure, basically. So we can then say that, like, if we're looking, well, ultimately we're looking to maintain the living root for as long as possible and as often as possible. And within that, basically, we would look to prefer annuals over perennials for leys and cover crops, and otherwise aim for what we might call the 5-minute fallow. Yeah. So go straight in with the next crop. So number 2 on soil health principles is to maintain a covered soil. Yeah, because by doing that, we're protecting soil and organisms, we're preventing capping, minimizing runoff, and promoting germination. So we look at rainfall simulator here just to illustrate the differences between different uncovered and bare soil. So that there's kind of 2 samples here, the one on the right and the one on the left. They're both identical in every way, same field, same day, same treatment, everything. The only difference is the one in the middle has had a light mulch applied to it, in this case some dried grass. Yeah. And by doing that, basically, what have we done? We've more than halved the amount of runoff. Yep. So just by covering the soil, we've massively increased the infiltration. Yeah. And one of the things that's really interesting when we finish this test, basically take the trays and you flip them over. And then one on the right-hand side, what do we think it looks like on the underside? Yeah. Even though they've already just pushed out about 2 inches of water in this sort of 20-minute time frame. Well, the answer is none, basically. That soil on the right is just bone dry underneath. So not just a bit of water, not just some water got in, but none. Yeah, it all ran off. Yeah, and when we add the living roots, the one on the left-hand side, for example, we have much improved and much cleaner runoff and much more infiltration, basically. So we can start to stack these different principles together, basically. So that's covered soil, basically. So what are we looking to do here? We're looking to cover soil with leaves and growing plants, for example, close planting, or with residues. So number 3 on our soil health principles is to minimize disturbance. Yeah, because by minimizing disturbance, we're preserving our aggregate structure. We're not chopping them up and opening them up to oxygen. We're also preserving the organic matter that's inside those aggregates. Yeah, we're preventing slumping following a tillage pass, and we're also, very importantly, minimizing weed germination. So if we look at the Slake Test, we can kind of, what's it called, get an idea of sort of the comparison here. So again, of how sort of tillage has an impact. So in the middle, we're seeing conventional tillage. On the left, we're seeing conventional no-till. On the right, we're seeing forest soil, basically. And in this case, basically, the Slake Test is indicating what this kind of the integrity of the structure of the soil is. So the aggregate structure. So if the soil holds together, basically, it means it's able to resist the forces that the water is exerting on those soils as it rushes into the pore spaces. Yeah, so dried soil samples into water, and then if it sort of basically slakes, if it collapses and crumbles down to the ground, basically, then it has very poor aggregate structure. So we can see we go into the no-till, and that's definitely improved that. But if we go to the forest soil, it's like, yeah, what? And, you can basically lift out that soil, dry it, and repeat ad nauseum, basically. Whereas the no-till soil, if you handle that, it just collapses, basically. So that's minimized disturbance, basically. So what do we mean here? What do we want to do here? We want to select low disturbance establishment techniques. We want to crimp or roll cover crops, for example, or mow cover crops to terminate them rather than tilling. We would prefer annual cover crops over perennial lays because they're easier to terminate. And I would otherwise look to strip till or just shallow cultivate. So the next one on our sort of list basically is to maximize diversity. Yeah, because as the diversity above ground, so the diversity below ground. Yeah, so if we look at a couple of examples here, so this is following a spring cultivation. Yeah, so on the right hand side, just a grazing rye and vetch, standard kind of two-way mix, if you like. On the left hand side, it's a high diversity mix, what you might call a cocktail cover crop. Yeah, so 11 species or so. And in that situation, which of these two soils would you rather, or would be easier to get a seed bed from? Which of these two soils would you rather plant directly into? Yeah, well, I think that the answer to that is pretty obvious. It's the one on the left, basically. Yeah. So we might ask this question then, can we go beyond herbaceous in our sort of plantings? Well, yes, we can. We can take our diversity knob and turn it up a bit more by integrating agroforestry, for example. So species of organisms that are associated with herbaceous plants are different from those that associate with woody plants. So where those sort of roots and mycelial mats overlap, you get like a nutrient bump. Yeah. But can we then go beyond diversity in our cash cropping? Well, again, yes, we can. Yeah, and we can introduce bi-cropping, strip cropping, catch cropping, relay cropping, all these different techniques, basically. Yeah. And can we go beyond the sort of 2-crop barrier? Well, again, yes, we can. And here's a good example from a market garden where they've taken the diversity knob and taken it all the way up to 11, basically. Yeah. And, you know, there's some really kind of well-looking crops in there, basically. Yes, they're definitely taking a hit on sort of like convenience and picking and all the rest of it, but they're saving so much and they're producing some very healthy crops, basically. And as well as that, they've turned this kind of sort of lifeless clay soil under the permanent pasture into 6 inches of deep, well, 6 inches of friable, dark topsoil, basically. Yeah. And they've done that by applying all the soil health principles, living root 24/7, mulch any gaps in the space, it's no dig, and it's sort of maximum diversity. Yeah. And it's all cash cropping. There's no cover crops or anything like that. Yeah. And this is a very challenging site as well. And I think that's it. I think it's worth mentioning. Very heavy clay, north facing, high rainfall with altitude. So if they can do it, anyone can do it, basically. So that's maximize diversity. So the next one we look at is to feed soils, number 5. Yeah, and again, we're all familiar with this phrase, feed the soil, not the plant, where we would ask the question, well, what actually eats stuff in soil? Has soil got a mouth and a digestive tract? No, it's the organisms that do. Yeah, and the other key point to take away here is that dormant organisms don't associate with crops. So here's an example of changing over from one crop to the next. So what's missing here? Plants. Yeah, and if there's no plants, there's no roots. Without roots, there's no root exudates. So there's no foods going into the soil, basically. So you've got a whole bunch of hungry organisms there. Yeah, and they'll be eating the sort of like the necromass, you know, the kind of plowed down roots and horns and stuff like that. Basically, that won't last very long, maybe a week or so. And then after that, what is there left for them to eat? And again, when we say eat, that means food. And what's food made up of? Some carbon-based compounds, basically. So this is when we come right back to the beginning and we can identify, you know, what's edible in the soil. Well, it's the aggregates, the stuff that's the polysaccharides, the stuff that's holding together those aggregates, basically. So if we allow this to persist too long, basically what will happen, these organisms will basically consume the stuff that is holding the aggregates together. And then what happens to the soil structure? It just slumps, basically. That crumb structure is lost. And then those organisms go dormant. So when it rolls around to you planting your next bunch of crops, and they go into the soil, start producing root exudates as they grow, silence, because all those organisms have gone dormant. Yeah. So what do we want to do here? We want to feed our soils with some kind of bulk organic matter, for example, between crops. Yeah. So if we can't ensure the living root, then we want to ensure that we're providing a subsistence ration between crops or over winter. Yeah. So next thing we talk about is number 6, is to incorporate animals, basically. And that's to say that like we can sort of integrate livestock and their manures, and they will then become part of the nutrient cycle, basically. So we've got the opportunity to incorporate them, particularly, I think, with cover crops, basically. So the final thing we'll look at then is the final number 7 of the soil health principles is to minimize chemicals and synthetics. And I'm not sure how many of you guys are organic here, basically, but the kind of take home here is that their use undoes your good work with the other principles. Yeah. And the kind of thing to say here is that when we apply a synthetic fertilizer, basically, it causes the plant to shut off the exudate pump, basically, because exudation is a quid pro quo for plants. It's a you scratch my back, I'll scratch yours. Here's some sugars, now where's my nutrients? Yeah. So when you fertilize a plant, it's like, I'm fine. That's it. I'm no need to produce exudates, basically. I'm well nourished, basically. And that's fine for that short period. Or then what happens over those sort of few weeks, basically, it then runs out of those, becomes nutrient deficient, basically. And then because it can't access the nutrients from the organisms, because the organisms have basically gone dormant, yeah, then the plant shows signs of chlorosis, at which point you then add more nutrients. Okay, so the what are our conclusions here basically? Is that most of the soil health principles, I'd say pretty much all of the soil health principles from myself, have been borne out through direct and ongoing observations. Yeah, and it's aggregation that correlates with these soil health practices. Where you see these practices in place, you see these structures basically. So let's come back to these this image that we looked at earlier basically, and compare these two soils in terms of the soil health principles and how they're doing for the soil health principles. So the one on the left, living root, covered soil, undisturbed, very much not. Diversity, probably not. No, basically, arable, maybe 3 or 4 crops in rotation. So low diversity and probably lots of chemicals. Yep. The one on the right-hand side, living root. Yep. 24/7, 365. Yep. Covered soil. Yep. Undisturbed. Yes, probably more than 10 years, I think, for this particular margin. Diversity, not too bad. Could do better. Chemicals, minimized chemicals, also pretty good, basically. Yeah. So is it any wonder that the one on the right looks in so much better heart than the one on the left? So this kind of brings the whole thing in full circle. That again, we all know that. So when your land goes to grass, comes out of grass, it's in better heart, basically. Yeah. So what this is about is ultimately enabling you to bring the benefits of grass without having to go to grass, basically. So what are we trying to do here? We're basically trying to mimic the kind of conditions we'd see under these kind of plantings, for example, in all of our cropping practices as much as possible and as often as possible. So just kind of final take homes here before we direct you to one particular case study. And that is that the goal here fundamentally is to turn underlying subsoil into fertile topsoil through the action of plant roots and microbes. Yeah, it all starts with observation. Aggregation is your primary measure of soil health, and it is easy to spot. Yeah, you don't need to send it off to a lab or anything like that. Yeah, and that fundamentally we want to let our management goals, let the soil health principles dictate practice rather than practice inform our decisions, basically. And that what we're trying to do here is identify those easy wins, the low-hanging fruit. And actually, it's accumulation of small wins that takes us along the journey, basically. So I'm just going to sort of direct you to these guys here and make sure that, kind of sort of recommend their work, basically. Singing Frogs Farm, they basically have applied the soil health principles rigorously and doggedly across all aspects of their work. And through doing that, basically, they've reduced their water usage by 90% and their water bill by 90%. They're now grossing over $120,000 per acre. Yeah. And they're maxing out at 5 to 6 crops per bed per year, where their neighbors are maxing out at 2, basically. So we can say that soil health is profitable fundamentally, as opposed to just like kind of do-gooder points, if you like. So your take-home messages here are that we should use, or I encourage you to use, the soil health principles to make decisions. Yeah, so in any given situation, those practices or interventions that score higher against the soil health principles should be preferred where practical. Yeah, and that basically the more the soil health principles are applied, the better the results. The fewer grade outs, the less moisture stress, the less disease, these kind of things, basically. And again, it's this idea of the accumulation of easy wins. So what are the things, what's your kind of homework? What would I like you to do this year, basically, is to just have a go. Yeah, and just dip your toe into some of these different practices, some of these novel practices, and have a go at intercropping one bed, or ideally 2 half beds. Yeah, so two split bed trials, for example, and the same with diversifying our cover crops. So if you're using cover crops or green manures already, great. Do half the bed with a diversified one, for example, a mixed one, and the same with try sheeting down couple of half beds and mulching a couple of half beds, and compare between the two. Is one a bit more slugged than the other, for example, or maybe won't use that again? You know, road test some of these different practices, but see how you can incorporate them more and more into your work, basically. And this is, these are the kind of like the sort of rules of the game, if you like. And these principles I use across the board with all the work that I do, whether it's client work, whether it's analysis work, whether it's rotation planning, whether it's education. These are what inform my thinking and my monitoring practices. So that's all from me. And we should now basically open up to the floor. But I might come out of the screen share to do that. So there we go. So if one of the moderators wants to drop in, that might be good.
Hi, Niels. We've got some Q&As.
I can deal with these questions basically. So just a very brief one. So someone asked, what is adaptive grazing? Not sure where you got that one from, but basically that is in the presentation. And basically we mean things like mob grazing, adaptive multi-paddock grazing, holistic planned grazing, these kind of techniques basically. Okay, another one from Laura Norman. In general, how long can tilled pasture be fallow before microbes start to eat exudates and seriously degrade soils? Days, weeks, months? Well, again, the question is, do we need to till that pasture first and foremost? So we'd all look at this in a hierarchy, basically. So if we can eliminate that tillage pass, then all the better, basically. But there's certainly a degree of necromass, you know, the roots and the tops, basically, that you would be tilling in, and that would last for a few weeks, basically. But the thing in that situation is you've degraded the soil often just through the process of tilling. Yeah. So we would look to like eliminate that, and one of the ways to do that in a mixed farming situation would be to move towards annuals rather than perennials, because we have that many more options for destruction, basically. But again, without knowing the detail of that particular question, yeah, I can't put more flesh on the bone, basically. But a lot of it would be dependent on the size of the area, for example, and also what the turnaround times are. So are we talking about sort of like this season, or are we talking about next season, for example? You know, if we're on a very tight turnaround, for example, we've got fewer options and tillage is much more indicated. Yeah, if we've got a longer time period, then sheeting and other options may be indicated, but only at a particular scale, obviously. Okay, so Jessica's got 3 questions. So number 1, what is the best way to terminate a ryegrass lay in order to plant in a no-till way more diverse cover crops? Well, I wouldn't terminate one cover crop to plant another, really, in this situation, because you just kind of, I would go straight into a cash crop personally, and a diversified cash crop potentially. Again, you know, depending on the scale, you know, if on a smaller scale, then sheeting down is indicated. On a larger scale, then obviously tillage is probably one of the only ways at an organic situation. But ideally, you probably would want to try and phase out ryegrass or perennial grasses or red clovers from the rotation, basically, and try and substitute those with diverse annual mixes or diverse combinations of annuals and perennials, biennials, I mean. So probably one of the examples that we didn't touch on the talk, as we come back to here, is some suggestions of cover cropping options. So let's see if I can find them. So one of them might be this one here. I'm not sure why you're not going, because I'm not pressing them yet. Selected. Excuse me, whilst I just find the one I'm looking for. So a couple of suggested practices. The first one is what I call successional cover crops. So mixtures of like tall straw cereals and biennials. This will give you kind of a 2-year planting. So the tall straw cereal can be terminated by rolling or flattening down at the end of the first year, and then the biennial sweet clover grows on for a 2nd year. And it's that much easier to terminate because it doesn't grow from a crown or a rosette like red clover does. It's just a single node upright. And the other option we would look at for sort of a single season, like a full season cover crop, would be cocktail cover crops. So these kind of diverse mixed species cover crops. So otherwise, crimping and rolling. So the other question is, Niels, have I had experience with crimping cover crops where it is wet and lots of slugs? No, not directly. But as with any mulch system, there has to be a transition period, basically. So we would target when that's done, and we would experiment with it, basically. But generally speaking, the experience is that slugs will decline when you move to a more soil health system. Certainly people like Ian Tolhurst report little or no issues with slugs. Obviously he's not in Wales, so that's like a difference in that situation. But I think the thing in those particular cases, they've got like beetle banks and areas to build up ground beetles, rove beetle populations to help to control slugs. But otherwise we might look at poultry and fowl to do that work for us, basically. But certainly kind of like I wouldn't sort of dive in blindly with high mulch systems, basically, without taking slug precautions or monitoring them. Yeah. So otherwise, I'm mentioning annuals over perennials, but what about compaction and the ability of deep-rooted cover crops, say chicory, to correct compaction? Well, I guess the first question is, where's that compaction coming from? Yeah, what's causing that compaction? Yeah, if it's your current management practices, which often is, then we should look to eliminate those management practices. We want to head those off at the pass, basically. We'd always look to work at the source of the issue rather than at the symptom end, if you like. Certainly, it's true that chicory has a sort of kind of robust spike root that can bust plough pans, yeah, but that's only over a period of time. You know, that's sort of 2 years and more, basically. So if your rotation has sort of 2 years or more, I would say probably for a herbal lay it's more like 3, because you don't really get anything in that first year, basically. It's only once you're into the second full season that they actually start to do anything, and then the 3rd year, that's when they really sort of start to rock, basically. That said, you know, we can still get big, robust and aggressive root growth from annuals. Yeah. In fact, probably the same, or if not more, just not so much in terms of spike rooting. But I think that kind of like differentiating on different rooting types is slightly overplayed. Yeah. Because even like a standard, sort of bog standard old ryegrass basically can grow a kind of like 3-foot long roots. Yeah. It's really the management that dictates that. So James, I think that might have sort of covered your question. So yeah, cocktail cover crops, successional cover crops are the kind of things to, as go-tos. But yeah, kind of experiment. And if you want to keep costs down with diverse mixes, one of the things you can do basically is to, let's see, come back here, is to use birdseed. Yeah, so deluxe birdseed mix contains pretty much all of what you need, basically. Nice diverse mix. Yeah, and you can also incorporate your own home saved seed, or those brassica seeds that are never going to get planted, for example. Just whack them all in. You know, really ramp up the diversity, because we want cheap diversity here, because then we can push the seeding rate up. Yeah. What's really important though, when you're sourcing birdseed mix from the feed merchant, is that you make sure it's not irradiated. Yeah, because they do that to stop it germinating off the bird table. You know. So if they either say, "Oh, don't know," or say yes, then go elsewhere, basically. But it's definitely you can find it elsewhere. Okay, so that's done. Cover crops. Not sure what Laura Norman was saying there. So what about AD plant liquid spread on soil toxins, plants and worms, etc., or not? Okay, so AD plant liquid. Not a product that I'm particularly fond of. personally. However, it definitely does contain nutrients and is often available in abundance. So I guess again, it's sort of a little and often is probably preferable over sort of a heavy dose. But I think often if it's spread by the contractor, they'll want to go heavy basically and get it out and get rid of it basically. If you can access AD digestate, particularly the stuff that's been squeezed or spun, that's totally dry, I think that's, well, from what I've seen, is an excellent mulch and soil conditioner. It's like straw, but with sort of compost in it, basically. But it's very fine. It's easy to mix and to spread, and it's very light, basically. So I think it will make quite a good, make an excellent mulch, and probably sort of low sort of attractiveness to slugs, basically. But this material is pretty new to me and to the market, so I don't know if it's available for you. Okay, so thanks for presentation. Have you any idea for keeping coop grass out of my organic system? It's becoming a problem each year. Well, going no-till is in its own right will make a difference, yeah, because couch grass is an early succession weed, basically. Its niche is competition-free environments, basically, where it can expand rapidly and occupy these new spaces. Yeah, and by the constantly tilling, basically, you're also making the sort of soil conditions more conducive to those types of early succession plants. Yeah, so I certainly know example from Jake at Oxton Organic, Jake Eldridge, where his mum basically was basically skeptical about going no dig. Yeah, they had been managing that particular holding, I think 4 acres, 5 acres or so, as a sort of like mixed organic horticulture operation for about 20 years, maybe 25 years. And she was very skeptical about them going no dig, and I think they went no dig in the tunnels first. And she's pretty much, she's going on record saying that right in the first year they got on top of their couch grass like they never did throughout the entire of the 20 odd years that they managed it beforehand. So I think kind of making these systemic changes brings kind of more kind of systemic solutions. That said, I think kind of incorporating like sheeting down, for example, making sure that you are using or preferring non-disturbance weeding techniques, for example, if you are using them, but certainly avoiding things like bastard fallows and all that kind of stuff, because what we want to be doing is growing really sort of like competitive, healthy crops, basically, that will compete with those plants. So yeah, I don't know if that answers the question specifically. But yeah, do check out Oxton's Organics. Jake contributes to the Organic Market Gardeners list on Facebook, and yeah, she was pretty like kind of, I'm impressed. I was impressed. I couldn't believe it. We did it in like the first year, and I've never done it in 20 odd years. But again, the thing I can't really attest to is the detail of what they did, except that I think it was the more kind of like council compost, deep compost mulch type things, for example, combined with sheeting down. So again, I'd need to know the scale that you're working on and more detail, basically. So the other one question: can these principles easily be applied to growing crops in pots or in polytunnels? Well, polytunnels, it's common. No dig is a bit of a no-brainer. It's uncontroversial in tunnels across the board. Basically, I would say that most growers manage their polytunnels using no dig, for example. Yeah, and that's basically because the value of the space is sufficient that it warrants that additional, or you can sort of subsidize that additional labor input, basically. So this is one of the things that we talk about in detail on the part 2 of my soils course, basically, is how we can bring the conditions that we find in polytunnels to the field. Yeah, kind of working back, if you like. but otherwise growing crops in pots, less applicable here, I would say. This is when you're going to be talking about producing, you know, good quality aerobic composts, for example, and using that as a medium or top dressing, working with worm liquids, for example, just worm extracts, or running wormeries, for example. In that situation, you know, it likes more sort of high nutrient, high input, basically. I haven't really got to worry about things like disturbance and weed growth so much, etc. So yeah, hope that's good. Yeah, if anyone else wants to drop any other questions in, I'm sure we could deal with them, but let's see what the other moderators have to say.
Oh, Niels, that was brilliant. Thank you very much. Thank you for your time this evening. It was fab. Slightly over time, but it was all really useful stuff. Just to let everybody know who's on the webinar, there'll be a recording on the Tyfu Cymru website through the Knowledge Hub, and I think there's Bethan's just put the website address in the chat, so please go there in the next few days. It won't be up immediately, give us a few days and we'll get it up on the Knowledge Hub. Just a couple more things before you go. When you leave the webinar today, there'll be a really short evaluation that pings up when you leave the webinar. There's about 7 questions on there. It will take literally a few minutes, and if you can complete that, we'd be really grateful. It just helps us shape how we can deliver more tailored training to you in the future. That's really useful. If you can, we'd be grateful. We do have lots more events coming up. There's lots of exciting things. Please go to our website. On the website, if you haven't already, there's a subscribe button. Put your details into that subscribe button and you'll get regular emails from us, just giving you an outline of what events are coming up. And I think that's it this evening. If anybody else has got anything to add, or are we all good? We're done. Thank you very much, Niels, and we'll speak to you soon. Thank you. Thanks for your time. Bye.
Yeah, thank you.
Everyone. Bye-bye.
Bye-bye.